The concept and proof of recombination events was introduced in our previous lesson. Let's now consider the steps involved in homologous recombination. As we saw in our last lesson, this process is employed as a means of repairing double-strand breaks in the DNA. In this process, homologous or like sequences are used in a recombination event. The Holliday model is used to describe how this event occurs, which involves the physical exchange of DNA. This process begins with an alignment of homologous DNA segments containing the same or nearly the same DNA sequence. First, the DNA backbone is nicked at a specific location, the same on two strands as shown below. Next, the strands are exchanged, shown here as an exchange between the blue and pink strands. As the two strands cross over at the nick, strand invasion occurs. The two strands begin to swap positions at that point. As branch migration proceeds, strand exchange occurs between the two DNA pieces. As you can see in the figure, the exchange only occurs from the nick downstream. As you can see from the center of the figure below, a quaternary complex forms between the four strands of DNA. Resolution of this Holliday structure is accomplished by cleaving two of the backbones. Cleavage on different strands results in either a patched recombinant on the lower left or a spliced recombinant on the lower right. Notice that in the patched recombinant, a segment is exchanged, but the rest of the DNA strand is identical to the original, both upstream and downstream of the patch site. In contrast, the spliced recombinant results in DNA pairs where each exchanged strand is a splicing of the two original strands. The Holliday model of recombination can only occur in cases where more than one copy of a gene or DNA segment exists on the chromosome.